Hi again. Just reminding you to give yourself this few seconds. Probably feels like you have a million things to do and you don't have time. It's worth it. It's worth it to let your mind settle in. Welcome back. Really glad you're here. As always, ecstatic you're here. We're going to continue our journey, our discussion, our understanding of what the heck is actually happening when your nervous system does something, when your nervous system fires, as we very often hear it described as. And as I said before, it's all about electrical communication. So now we're going to set the stage for how does that electrical communication happen? What is even going on? So electrical communication, what it is, electricity, is the movement of charged particles. And of course, we could get far more technical than this, but this is what we're going to focus on when we're discussing the immune system, is that electricity is the movement of charged particles. So if we're looking at our neuron, as clumsily drawn by me, the focus of understanding the physiology of this, the nervous system, is understanding this electrical signal and how it's propagated. So as we talked about before, it comes from some signal from the dendrites down the axon to the terminus. And this electrical signal, this electrical change in the neuron, stimulates the release of neurotransmitters. I'm just going to remind you of a really important thing that we can't forget in any of our discussions about any of this, is that this target cell, which I've just drawn as like this sort of box here, we're talking right now about the target cell being another neuron, but it doesn't have to be. It can be an endocrine cell. It can be a muscle cell. It can be so many different things. So this target cell, whether it's a neuron, whether it's any kind of cell in the body, has to have something for any of this to matter. This target cell has to have what? All of this, everything that we're talking about, isn't going to matter a single solitary lick if this target cell doesn't have what? What does it have to have? Target cell, boom, has to have a receptor, right? It has to have the right receptor. So this target cell, whether it is a neuron or any other kind of cell, has to have the right receptor for the neurotransmitter that's being released from that neuron. So all of everything that we're going to talk about to produce this electrical signal, the goal is to release a neurotransmitter into the synaptic cleft at a target cell that has the correct receptor. If it doesn't have the correct receptor, we can release all the neurotransmitter in the world, and it's not going to matter. That target cell ain't going to do anything. Keep that in mind. Okay, so as I said, electricity, movement of charged particles. And so specifically in the nervous system, this electrical signal that's propagated down the length of a neuron is called an action potential. Action potential. Action potential is when that electrical signal gets propagated down the entire length of the neuron. So the whole neuron fires. Okay? That's the action potential. We're going to talk about a different kind of potential in a bit. So the action potential is when that electrical signal is carried down the entire length of the neuron. Okay, how the heck do we get that? How do we get an electrical signal being carried down the length of a neuron in our bodies? Every time I teach this, no lie, and I actually recorded this lecture before and thought my stuff was working and it wasn't, so I've just said this very recently. It still blows my mind that me raising my hand, me yapping my jaw, you perceiving this lecture right now is all based on electrical signals. Electricity in your body. It's so cool. Okay, how does that happen? Well, how that happens, we have to have the movement of charged particles to get electricity, right? So how that happens is we have to have charged particles. We have charged particles and a difference in charged particles inside the neuron versus outside of the neuron. So this is my neuron drawing. And along the middle, we have these red dashes. And those red dashes, what those indicate is they indicate a negatively charged environment. And that negatively charged environment is in comparison to the external environment, which is positively charged. So if we compare the charges inside the neuron to the charges outside of the neuron, inside is more negative. So it's relative, right? We can't just say it's negative. It's negative in comparison to what? It's negative in comparison to the fluid surrounding the neuron. So the internal environment is negative, external environment is positive. And I just revealed the answer already. But before we get to that, this negative internal environment, positive external environment, what is this called? When we have a difference, we could have a difference in charge, we could have a difference in dissolved solutes. What's that called? That's called concentration gradient. It all fits together. Okay, this is a concentration gradient. And you've learned through lecture and through your experiments in lab that nature wants to balance this. Our bodies or nature wants to, I'm anthropomorphizing, the natural movement of our body's particles leads to a balance in these things. It leads to the loss of a concentration gradient. But so much of the important physiology of our body is driven by concentration gradients. So what that means is that our bodies have to actively work to establish this concentration gradient so that a neuron can fire. And the star of that show, my dog is not the star of that show. My dog was just making noise. The star of that show is the glorious and fantastic sodium potassium pump. Sodium potassium pump. And I know that you've learned about the sodium potassium pump, at least in introductory biology. It's so wildly, critically important. Sodium potassium pump. Okay, so sodium potassium pump, you'll also see it written. Sodium is Na, potassium is K. So you'll see it written as the NAK pump. Another way you'll see it written is as the sodium potassium ATPase pump. All of these are referring to the same thing. Okay. So the sodium potassium pump is the star of this show, really. We glorify the neuron, like the neuron is out there doing its thing, but it cannot do its thing without the sodium potassium pump. What the heck even is the sodium potassium pump? Okay. Remember from our discussion of the electron transport chain and our discussion of the cell membrane that the membrane is impermeable to most things. It's semi-permeable membrane, but most things can't just get across the membrane. So these little green structures with tails, they're representing the phospholipid bilayer. And if you'll remember, specifically from when we talked about the electron transport chain, that embedded in that phospholipid bilayer are proteins. All sorts of proteins embedded in the phospholipid bilayer, right here. And they span the phospholipid bilayer. So one end of that protein is in what we call the extracellular fluid, so outside, and another end is in the intracellular fluid. So they're like a door. Okay, so these proteins are like a door or a hallway through that phospholipid bilayer. Okay, so we've got proteins embedded in the phospholipid bilayer. The sodium-potassium pump is one of these proteins, one of these super cool, specialized, amazing proteins. And the sodium-potassium pump is the type of protein that it's only ever open to one side. So it's not a channel, and we'll talk a lot, we'll have a lot of channels involved in this discussion. It's not a channel that can be open to both sides at the same time. It's only open to one side at a time. So I kind of mentally, I think of it as like this little Pac-Man where it's open to one side at a time, closes, opens to the other side. So in this drawing, here is the extracellular fluid and here is the intracellular fluid. Oops, that's not right. Go away. Here's the intracellular fluid. So the top is outside of the cell and the bottom is inside of the cell. And the yellow line is, I've just simplified my drawing here of the phospholipid bilayer so I don't have to draw all those little heads and tails. Okay, so we start out with our sodium-potassium pump, and this is a continuous process. So I'm starting it open to the intracellular fluid. You could just as easily start it open to the extracellular fluid. Either one, no worries. So we've got our protein, our specialized protein, and right now it's open to the intracellular fluid. It's open to the ICF. And when it's open to the intracellular fluid, it's in a shape, it's in a configuration where it has 3 binding sites for a very specific ion. It has 3 binding sites for the ion sodium. Sodium is positively charged. So it opens to the intracellular fluid. It has these binding sites. So what happens is that 3 sodium molecules come and they bind. Okay, we get 3 sodiums that come and bind to the sodium-potassium pump. Cool, right? The sodium-potassium pump, what this does when these 3 sodium binds is it causes a conformational change. So it causes this protein to change shape. And this is a really common thing, is that, remember, the protein, I'm just drawing it as this sort of like Pac-Man-y thing here, right? But it's this incredibly complex structure. So over here, I've drawn a very slightly more accurate version of maybe what a protein would look like. So remember back when we talked about important biomolecules, we talked about protein. And protein has primary structure, secondary structure, tertiary structure, and sometimes a quaternary structure. And in each of these structures, we get more complex and more folds. So if we think about this incredibly complex 3D folded structure, and what we've done is we've come and let's just imagine our binding sites are here. And those, when it binds, those binding sites cause this complex structure to change shape. So maybe it just rotates a little bit. Maybe a part that was tucked inside gets pushed outside, this conformational change. And when it changes its 3D structure like that, what it very often does is it reveals new binding sites. So binding sites that were hidden, binding sites that didn't have access. Now we've changed shape, and boom, there's access to a new binding site. In this case, that binding site is for ATP. Okay, so we're binding ATP. ATP is cellular energy. If we're binding ATP, we're going to use ATP. So what kind of process is this? Is this an active process or is this a passive process? What is this process? Active or passive if we're using ATP? It's an active process. We're having to use up some of our cellular energy. Okay, so we bind ATP. Cool, cool. So we've bound sodium, we've bound ATP, and then what happens is that ATP is hydrolyzed. Hopefully that word sounds familiar. And one of the phosphates gets broken off. Hopefully you remember that when one of those phosphates gets broken off, that releases that potential energy in those bonds. So a phosphate gets broken off, it releases energy. So the phosphate stays here, and this releases ADP. Hopefully this process sounds pretty familiar. Okay, so now what happens is that once again, our protein changes shape. And in changing shape, what it does is it opens itself to the extracellular fluid, to the extracellular environment. And in so doing, it releases its sodium into the extracellular environment. Okay. So we release our sodium into the extracellular environment. We create now new binding sites, or reveal new binding sites. Those binding sites are for another ion called potassium, sodium-potassium pump. So now potassium comes in and it binds its two binding sites. And potassium binds, and we get a shape change, and we get the release of that phosphate gets released. Is that the right color? The phosphate gets released, and the potassium gets released into the cell. Okay. So this whole process where we're conformationally changing, we're moving sodium to potassium, we're using cellular energy, that cycle happening over and over and over and over again. What it does every cycle is it pumps 3 sodiums out of the cell and 2 potassiums into the cell. So that probably, I don't know, maybe to you, you're like, okay, cool. But for me, I was always like, why are we doing positive and positive? Like, that doesn't make sense to me. But think about it this way. If we were having in-person class, and every time you walked into lecture, you had to give me $3. And I always gave you $2 back. Oh, thanks for that $3. Here's $2 back. Which one of us walks out of that lecture with more money? I do. I gave you $2, but I netted 1, right? So even though we're pumping positive into the cell and positive out of the cell, the extracellular fluid is going to become more and more and more and more and more positive. Because remember that this sodium-potassium pump is just working all the time. And so this is how we establish, oh, did I do those colors backwards? I did, guys, I'm sorry. I did those backwards from my earlier indications of what was happening with the positives and the negatives. So over time, we get more and more positive extracellular fluid and more negative intracellular fluid. I apologize for flip-flopping on you with the colors. Through the action of the sodium-potassium pump, which is primarily what establishes this. There are some other important things involved. The distribution of proteins on either side of the cell membrane is also important. So the entire balance of everything. But the sodium-potassium pump is a key, probably the key, to establishing this positive on the outside, negative on the inside. I am so sorry that I flip-flopped that on you guys. I think that could be very confusing, couldn't it? So let's see if I can fix that. Ah, no! Okay, so positive on the outside. So that's more sodium on the outside. And negative on the inside. Okay. All of the facts were the same. I had just flip-flopped the colors, and I did not want that to be a source of confusion. Okay, so this difference in charge, the difference in charge across a membrane, is called membrane potential. So this is the difference in charge across a membrane. Is across two C's? Across. I'm so bad at that. A membrane. Okay, this is the membrane potential, and that's because this is potential energy. So these ions have the potential to move. And in our neurons, we have something called resting membrane potential. And resting membrane potential is what's the difference in charge that our neurons just sort of hang out at, the homeostasis that our body tries to maintain. How different is that charge? And the resting membrane potential for our neurons is about negative 70 millivolts. So the inside of the neuron is about 70 millivolts more negative than the outside of the neuron. So this is just where our homeostasis is. This is our resting membrane potential is negative 70 millivolts. And I promise I'm telling you that for a reason, and we'll get to that later. Okay, so that's the sodium-potassium pump. That's how this difference in charge, this resting membrane potential, gets established. Next, we'll talk about how do we change that? How do we get electricity? How do we get the movement of charged particles?